Hello students, welcome to zoology class. Hope all of you are fine at your places. Today, in continuation to my presentations on cell structure and function, and we have started with the different cell organelles, and today the next organelle which we will discuss, that is centrosomes and centrioles. So today we are going to discuss about centrosomes and centrioles. So what is centrosome and then centrioles? The term centrosome, it was given by Theodore Boveri in the year 1888, and these centrosomes, they are present in all the animal cells, but they are absent in most of the plant cells. And the centrosome, it is made up of two perpendicular centrioles, a daughter centriole and a mother centriole. And these two centrioles in the centrosome, they are linked together by interconnecting fibers. And centrosome consists of a complex of proteins which help in the formation of additional microtubules and an amorphous pericentriolar matrix. It surrounds the centrioles, and they are also known as diplosomes. So I will tell you that the centrosomes, they are made up of two perpendicular centrioles, means inside the centrosome there are two centrioles, and these two centrioles, they are present at right angle to each other, that is they are perpendicular to each other, and one centriole is the daughter centriole and the other centriole is the mother centriole. And these two centrioles in the centrosome, they are linked together by the interconnecting fibers, and these centrioles consist of a complex of proteins, and those proteins help in the formation of additional microtubules during the process of cell division. And now you know that in the centrosome there are two centrioles present perpendicular to each other or at right angle to each other, and these two centrioles, they are surrounded by a pericentriolar matrix. Pericentriolar means surrounding the centrioles there is present an amorphous material. So these centrosomes, they are also known as the diplosomes. So now we'll come to the structure of each centriole in the centrosome. When we talk about the centrioles are cylindrical, rod-shaped microtubular structures, and they occur in a pair near the nucleus. And the two centrioles, as I already told you, that they are present at right angles when they are close to each other. And this pair of centriole is contained in a structure called centrosome near the nucleus. And the structure of the centriole is an array of 9 groups of triplet of microtubules equally spaced around the perimeter of the imaginary cylinder, and the space between and immediately around the triplet is filled with the amorphous electron dense material. And the centrioles, they lack limiting membrane, DNA or RNA, and the centrioles are found in pairs, but they move towards the poles or the opposite ends of the nucleus during the cell division. Now to further explain the centrosome and the centrioles, I will tell you that each centriole is a cylindrical rod-shaped structure, and it consists of microtubules. And this centriole or a pair of centrioles occur near the nucleus. And as I already told you, that as they are present near the nucleus and they are present at right angle to each other, and a pair of centriole is surrounded by amorphous material in the centrosome. So this forms the basic structure of the centrosome. So I mean to say that centrosome is consisted of two centrosomes and they are present near the nucleus. And if we see the structure of each of the centriole, it consists of 9 groups of triplets of microtubules, and they are equally spaced around the perimeter, that is surrounding the centrosome. And it is if we consider an imaginary cylindrical shape, then at the perimeter there are present 9 groups of triplets of microtubules in each of the centriole. And centrioles have no membrane, they have no DNA or RNA, and centrioles are present close to each other near the nucleus, but during the cell division, this pair of centriole, it moves towards the opposite ends, that is opposite poles. So this is the basic structure of the centriole. So centriole is made up of 9 sets of microtubules, and they are arranged in a circle like the cartwheel organization, and these microtubules, they are tilted at an angle of 40 degree, each in groups of 3, which are known as triplet microtubules, and the arrangement is known as 9 plus 0 because there are 9 groups of triplets at the perimeter, and in the center there is no microtubule. And triplet microtubules, they are very strong because they are composed of 3 concentric rings of microtubules which are connected together. And triplet microtubules, they are seen in other strong microtubule structures such as the basal bodies of cilia and flagella. And each triplet is bonded together by special proteins which give the centriole its shape. And surrounding the triplet microtubules is an amorphous material and which is contained in the centrosome. So each microtubule is a triplet and it is made up of 3 subunits, and these subunits are made up of the protein known as tubulin, which is a small monomer, and they can join together to create long hollow tubes which resemble the straws. So this is the structure of the centriole. So if we see the diagram, so this is the centrosome, and as I told you that centrosome is made up of 2 centrioles. This is mother centriole and this is the daughter centriole, and these 2 centrioles, this is one centriole, this is the other centriole, and they are present at right angle to each other or perpendicular to each other. So this is the structure of the centriole. Then if we further explain the structure of the centrosome and the centriole, so this is the cylindrical shape, this is one centriole and this is the other centriole. And if we study each centriole, as I told you that each centriole, it is made up of 9 groups of microtubules, and they are arranged in a circle like cartwheel organization, and they are tilted at an angle of 40 degree. So this is 1, 2, 3, 4, 5, 6, 7, 8 and 9. So these are the 9 groups of triplets and they are arranged in a cartwheel like structure. So this is the cylindrical shape, and there are 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is shown here. So these are the 9 triplets and they are arranged at an angle of 40 degree, and these microtubules are made up of, these are the microtubules, and each triplet, this is 1, 2, 3. So these are made up of the protein known as the tubulin. Then so these are the 9 units and the periphery. So this is the periphery. So this is the perimeter of the centriole. These are present at the periphery, and there is no protein in the, this is proteinaceous hub, but there is no triplet or there is no no unit in the center. So it is known as 9 plus 0 organization. So this is the structure. So each of the 9 sets of the triplet composed of 3 subfibers. This is 1, 2 and 3. So each is made up of the 3 subfibers, and these 3 subfibers of the triplet, if we start from outside to inside, they are designated as C, B and A. So these are the 3 subfibers, and each subfiber, if we talk about C or B or A, each is made up of the 13 proto filaments, and both C and A, these share 2 or 3 protofilaments in B fiber. Suppose this is a B fiber, here it shares the microfilaments or the protofilaments of C and A. So this is the arrangement of the protofilaments in each triplet, and the adjacent triplets are connected to each other by proteinaceous linkers. They connect C of the one triplet to the A of the other triplet. So this is the linker. This red one, this is the linker between the one triplet and the other triplet. So it joins the C of one triplet to the A of the other triplets. In the same way, C of one triplet and A of the other triplet. Then again C of the one triplet and A of the other triplet. So these are known as the linkers. So the center of the centriole is occupied by the proteinaceous hub. So this is the proteinaceous hub, and each triplet is connected to this proteinaceous hub by the radial proteinaceous strands. So these are the proteinaceous strands which connect the central proteinaceous hub with the triplets, and these are known as the spokes. So this is the spoke. So okay, so this is one spoke, this is second, this is third, this is 4, 5, 6, 7, 8, 9. So these are known as the spokes. So the spokes are also connected to C and A linkers by 2 types of thickenings, and they are known as X and Y thickening. So these are the thickenings. These are the X and Y. This is X and Y thickening. So by the help of X and Y thickenings, these spokes, they are connected to the triplet C and A. So this is the organization of each centriole. So now you know that centrosome is made up of 2 centrioles present perpendicular to each other or at right angle to each other, and surrounding the centrioles there is present amorphous material. These are also known as the diplosomes, and they are present near the nucleus in the animal cells, and during the cell division process these centrioles move towards the opposite ends and it forms the more microtubules. So and these are each centriole is made up of the 9 triplets, and they are arranged in a cartwheel like organization, and this is the 9 plus 0 organization, and this is all about the structure of the centrosome. Next comes the function of the centrosome. Now you know the structure of the centrosome, so now comes the function. So major function of the centrosomes are that the centrosomes, they help in cell division and they maintain the chromosome number during cell division, because during the cell division these centrioles, they move towards the opposite end and they form the microtubules which maintain the number of the chromosomes during cell division. They also stimulate the changes in the shape of the cell membrane by phagocytosis. And in mitosis, mitosis is the process of cell division, and it helps in organizing the microtubules, ensuring the centrosomes are distributed in each daughter cell. So during the process of the cell division, the centrosomes, it helps in the distribution of the chromosomes in each daughter cell by forming the microtubules, and they also regulate the movement of the microtubules and cytoskeleton structures. So they facilitate the changes in the shape of the membrane of the animal cell. So this is the basic function of the centrosome, that they form microtubules during the process of the cell division, and they maintain the number of the chromosomes during cell division. So we can say that the centrosomes help in cell division. So these are the functions of the centrosome. So this is the structure showing the position of the centrosome during cell division. In actual shape, the centrosomes are present near the nucleus, but during the cell division, one centriole is here and other centriole is here. So they are attached with the these mitotic spindle fibers of the chromosomes, and they move towards the opposite poles during the cell division, and they help in the process of the cell division. So this is the function of the centrosome. So today we have discussed about the structure and the function of the centrosome along with the centrioles. So you have to study this presentation. I have already sent you study material, and then you have to prepare notes by studying both the presentation as well as the study material. And if you have any difficulty, then you can come to me for discussion. Thank you.